This video is on the immune system, part 3. So what we're going to talk about next is the cells of the specific immune defense. Now remember, we have already said that the immune system consists of two divisions, the nonspecific and the specific. So there are three important cell types in the specific defense. Two types of lymphocytes and something called an antigen-presenting cell. Let's start with the lymphocytes first. Well, you know that lymphocytes originate from hemocytoblasts in the red bone marrow, along with all other white blood cells. Lymphocytes have to be able to recognize a specific antigen by binding to it. And if they do that, they call this immunocompetence. Okay, so again, the ability to recognize a specific antigen by binding to it is called immunocompetence. And lymphocytes that are immunocompetent have a unique receptor that only responds to one antigen on one pathogen. If you looked at an immune cell, a lymphocyte, you would see that it has many receptors, as many as 100,000 receptors on the outside of the cell. But the receptors are all identical. They're all for that one antigen. So how do B cells and T cells, B lymphocytes or T lymphocytes, how do they become immunocompetent? Well, the first thing is, the first place they do that is in primary lymphoid organs. So remember, these are organs of the lymphatic system, and we talked about those in the very beginning of this chapter, things like the bone marrow, the thymus, the spleen. But the organs where lymphocytes become immunocompetent are either the bone marrow or the thymus. So if a lymphocyte becomes immunocompetent in the bone marrow, they call it a B cell, B for bone marrow. If the lymphocyte becomes immunocompetent in the thymus, they call it a T cell, T for thymus. So the process where this happens is fairly complicated, and I'm going to try to keep it as clear as possible. So let's take a look at this next slide that might help a little bit. So here we have the lymphocytes in the bone marrow. Can you guess what bone this is supposed to be? Well, if you guessed femur, you're correct. So remember, in the bone marrow, in this case they're showing it in the medullary cavity, are where all white blood cells and red blood cells are formed. But here we have two lymphocytes. So if the lymphocyte migrates or stays within the bone marrow, then it becomes immunocompetent there, and it's called a B cell. If the lymphocyte migrates to the thymus and becomes immunocompetent there, it's called a T cell. Now, once cells are immunocompetent, they travel to lymph nodes, the spleen, or other lymphoid tissue. And in this case, they are immunocompetent but naive. And what that means is they have the one antigen, which will help them recognize and bind to that, they have the one receptor that will allow them to bind to that one antigen that they're targeted for. But they're naive, which means they've never actually met the antigen. They've never met the pathogen. So they go to the lymph node, the spleen, or other lymphoid tissues. Often they're in the lymph node, and they wait until the pathogen comes along. When the pathogen comes along, they recognize it by binding to it, and now they are considered immunocompetent but mature. Okay, so once this happens, again, they are immunocompetent, but they are going to be mature. That means that they have the one receptor to bind to the one antigen, and they've actually met the antigen, so they are mature. And now what they do is they circulate throughout the body, throughout the blood vessels, looking for and hunting down that pathogen with the one antigens. So, a couple of things here. First of all, we have in our body about 10 trillion different B cell receptors. So in our body, we have 10 trillion different B cell receptors, and each receptor is specific for a particular antigen on a particular pathogen. We have 1 billion T cell receptors, but when you look at the genes available to make these receptors, there's only like 25,000 different genes. So how can 25,000 different genes give you 10 trillion different types of B cell receptors and 1 billion different types of T cells? Well, it happens through genetic recombination, which you might remember from biology, where the genes rearrange during the maturation of the T and B cells, and it's random. So if you randomly assemble 25,000 different genes, you can come up with a huge number of different receptors. And that's how we have receptors for almost every pathogen on the planet. Even COVID-19, we have receptors for those. We have B cells and T cells. The other thing I want to mention is the process that takes place when cells are becoming immunocompetent. So remember, the immunocompetence is happening here in the thymus and the bone marrow, and you have a bunch of cells. So think of these cells as, new students in a classroom, and they all want to become T cells. They want to become immunocompetent naive T cells, right? Or immunocompetent B cells. So we don't know as much of what goes on in the bone marrow, but we do know what goes on in the thymus. And in the thymus, these cells are exposed to 2 tests. And the first test is called positive selection. And in positive selection, the lymphocytes are presented with the MHC complex. Now remember, MHC complex are the group of proteins found on all your cells that identify your cells as self to the immune system. So the first thing that happens is they are presented with the MHC complex. And if they recognize it by binding to it, they pass the test. If they don't recognize it, they fail the test and they are destroyed through the process of apoptosis, which remember is programmed cell death. Then the ones that are left are given another test. The second test is called negative selection. And in this case, they're presented again with the MHC complex. But if they bind to it too tightly, then they are eliminated. And the reason for this is if they bind too tightly, the concern is that they will actually see your body cells as being foreign and destroy it. And this results in something called autoimmune diseases. So the second test is they can't bind to it too tightly. If they do, they're eliminated through apoptosis. And this is the type of testing that is going on within the thymus and the bone marrow. So when these T cells are being trained here to become immunocompetent, they don't all pass the test. In fact, you might wonder how many of these lymphocyte students pass the test to go on to become immunocompetent and mature, and only about 2%. So only about 2% pass the test. So it is a rigorous test. It requires lots of resources, but it's a way to ensure that the body does not circulate T cells and B cells that are going to damage your own cells. Now, one last point I want to make about this process is that these lymphocytes are becoming immunocompetent before they even meet the pathogen. So remember, they meet the pathogen in the lymph node, but they're becoming immunocompetent here. So in other words, they're acquiring that one receptor for a very specific antigen before you've even been exposed to it. So the question here is, what determines what our immune system reacts to? What determines what our immune system reacts to? And you could say, well, it's what you're exposed to. But it can't be, because remember that you have the receptor for the antigen before your immune cells are exposed to that pathogen. And actually the answer is it's our genes. It's in our DNA. So through the process of evolution, humans have acquired these genes that code for specific receptors for specific pathogens on the thymus and bone marrow. In the T cells and the B cells. And those organisms that had the genes for those receptors against specific pathogens, those organisms survived, and those that didn't survive. And that's how we have acquired over time these genes. Okay, so we said these are the cells of the specific defense, and we have two, the T lymphocyte and the B lymphocyte. But there is one more group of cells that is really important called antigen-presenting cells, or APCs. So antigen-presenting cells do just what their name suggests. They present the antigen to the B cell or the T cell. So again, their role is to engulf foreign particles and present the antigens on their surface, in this case to T cells. Major antigen-presenting cells include something called dendritic cells, macrophages, and B cells. So I just wanted to show you this picture of a dendritic cell. This is with a scanning electron micrograph. But these are large cells with these huge extensions, and they are present at the boundaries of your body. So your skin, mucous membranes, and their job is to engulf and destroy pathogens. Then they will take an antigen and stick it on the outside of themselves. They'll migrate to lymphoid tissue and present the antigens to T cells. And this is probably the most important way that our immune system is alerted to the presence of foreign pathogens. The major way is through these dendritic cells. Now, the T cells will often stimulate B cells, and then the B cells can present the antigen to other B cells. So let's watch this quick video here about how the cell responds.
Activation of the immune response typically begins when a pathogen enters the body. Macrophages that encounter the pathogen ingest, process, and display the antigen fragments on their cell surfaces. Macrophages with antigen fragments displayed on their surfaces are called antigen-presenting cells. An antigen-presenting macrophage interacts with a T helper cell that can recognize the same antigen. During the interaction, the macrophage releases a chemical alarm signal called interleukin-1, which stimulates the T helper cell to secrete interleukin-2. Interleukin-2 causes the proliferation of certain cytotoxic T cells and B cells. The immune response from this point follows two paths, one using cytotoxic T cells and one using B cells. Normal cells of the body that become infected can also digest some of the pathogens and display antigen fragments on their cell surfaces. The body makes millions of different types of cytotoxic T cells. Each type is able to recognize a particular antigen. The cytotoxic T cells that are capable of recognizing the antigen displayed on the surfaces of infected cells bind to the infected cells and produce chemicals that kill the infected cell. Death of the infected cells results in destruction of the pathogen. B cells also come in millions of different types, each able to recognize a particular antigen. When B cells become activated by T helper cells, they differentiate into plasma cells. These plasma cells become antibody-producing factories, flooding the bloodstream with antibodies that can bind to the antigen involved in this infection. Antibodies bind to the antigens on the surfaces of the pathogens, marking them for destruction by macrophages. Some of the B cells do not turn into antibody factories, but instead become memory B cells that may survive for several decades. Because of these memory B cells, the secondary immune response to a future infection by the same pathogen is swifter and stronger. This powerful secondary immune response is what gives immunity to some diseases after you have had them once or after you have been vaccinated.
Okay, so the purpose of that was really to show you how antigen-presenting cells work. They basically engulf the pathogen, chop it up, and take a piece of it and stick it on the outside of themselves, which they then present to T helper cells. Now, we're not going to have time to get into T cells and the details of how T cells work, We briefly said that they can either lyse the cell by releasing chemicals like perforins, or they can call in different parts of the immune system. But there are T helper cells which then activate B cells. So that kind of leads us into the humoral immune response. Now remember, there are two divisions of the specific defense. There's the humoral and the cell-mediated. So the cell-mediated response is pretty much directed by T cells. But the humoral immune response is really all about antibodies. So B cells make these antibodies, and the antibodies provide the protection. Remember that antibodies target bacteria, their toxins, and free viruses. We talked about this in an earlier video. And the reason they do that is they're just proteins in the plasma, so they can't get at pathogens that are inside a cell. They can only attack things that are outside the cell, which are things like bacteria, toxins, and free viruses. Their mechanism of action is to inactivate pathogens and mark them for destruction by other parts of the immune system. So, how does this humoral response take place? Well, the first thing is you have these B cells. And so the first challenge to the B cell by an antigen is called the antigen challenge. It's the first encounter between an antigen and a naive immunocompetent B cell. What happens next is that the B cell is going to clone. So the easiest way to show you this is with a picture. Okay, this is called clonal selection, but it's what happens in the humoral response. So at the top, you'll notice it says primary response. This is the first time someone is exposed to a pathogen. Here, you'll see 3 B cells. So these are supposed to be B cells up here. And you'll notice they have different colored receptors. So this one has green receptors. Maybe this receptor is for an antigen on the chickenpox virus. Here's another B cell with purple receptors. Maybe these ones target an antigen on the influenza virus that causes the flu. And then lastly here, you have another B cell with these blue receptors. Maybe these target the measles virus. So they all target different things. Well, along comes the flu. You're exposed to the virus that causes the flu. And here's an antigen. It could be presented to this B cell by another B cell, or maybe by a T cell. But you'll notice that it only binds to the B cell that has a receptor for that particular pathogen, which is the influenza virus. These little orange things are the antigens for the influenza virus. The other B cells don't do anything because they don't target the influenza virus. So immediately this B cell forms clones. Now remember, a clone is basically an identical cell. So the B cell is going to produce a clone, identical cell, and it makes lots of clones. The clones are going to make plasma cells, and the plasma cells make antibodies, these substances, that are shaped kind of like a Y, because that actually is the structure of an antibody. The other B cell is going to form a memory cell and maybe make some plasma cells. The memory cell is going to stay in your body for a long time, maybe your entire life. So the first time you're exposed to a pathogen, the primary response, the B cell that targets that antigen on that pathogen makes clones, which make plasma cells, which make antibodies. And the antibodies are going to fight off that pathogen. Now keep in mind that T cells are also targeting this particular pathogen, as well as the non-specific defense. So then let's say 2 years later, you're exposed to the flu again. See, years later. Now what happens? Well, you have that memory cell that already has receptors. It's already been trained. And so the memory cell immediately makes plasma cells and clones and plasma cells and memory cells. The plasma cells make antibodies. But you'll notice there's a difference between the secondary response and the primary response up here. Do you notice the difference? Well, you might notice there's more plasma cells and there's more antibodies, and there are. So the second time you're exposed, because of this memory cell, your response is stronger you produce more antibodies. This is a great video about the clonal selection, and I really recommend you look at it. I'm not going to go through it right now, but it's very, very good. But let's go on to this primary and secondary humoral response, and we're going to look at a graph here. So on the graph on the x-axis is time in days. On the y-axis is antibody titer. Titer means concentration. So what's the antibody concentration in your blood? How many antibodies are you making? And you'll notice that this is in the log to the base 10 scale. So remember that in the log base 10 scale, the exponent, the 0, means how many zeros do you have? So here it says you have 0 zeros, so this means 1. Here it says you have 1 0, so this is 10. Here you have 2 zeros, so 100. 3 zeros, 1,000, 10,000. Okay, so that's the scale that this is in. So let's look at the primary humoral response, which is shown in blue here. And you'll notice that here you're exposed to the influenza virus. But there's a lag time here before you start cranking out those antibodies. Lag time of maybe 3 to 6 days. Then you start making the antibodies, they rise. You don't make a whole bunch. Hopefully they fight off the pathogen, and then they slowly go back down. So this is the primary humoral response the first time you're exposed to the pathogen. Now I know this says 28 days later, but let's make believe, which is also a scary movie, but let's make believe it is 2 years later. You're exposed to the same pathogen, the influenza virus again, the flu. Now you'll notice you start cranking out those antibodies a lot faster. It's not 3 to 6 days, it's probably within a couple of hours. The other thing you might notice is the slope of the antibody production. You produce antibodies a lot faster and a lot more antibodies. Now you're up to almost 10,000 antibodies. The third thing you might notice is that the antibody concentration stays higher longer. See that? Whereas the first time you're exposed, it went down pretty quickly. But here it stays much higher longer. So again, this is the primary humoral response the first time you're exposed, and the secondary humoral response the second time you're exposed. Now this is supposed to show if you were exposed to something else, like the COVID-19 virus, you're still going to have the same kind of slope for a primary humoral response, because it's the first time you're exposed to that virus. So let's compare on this page here, and you may not have this slide. If you don't, please write it in. But this is showing us, again, the difference between the primary and secondary humoral response, which we just went over. So the first time an individual is exposed, the lag time, I think we said, was like 3 to 6 days before the body starts producing antibodies. But the second time you're exposed, the lag time is within hours. It is a lot faster. Sorry, that's supposed to say faster. So the response is faster. Number of antibodies produced, the first time it was less. The second time it was more. So we could say the second time you're exposed, the response is stronger, right? And then in terms of concentrations of antibodies, it drops fairly rapidly, right? Maybe 4 to 5 days, something like that. The concentration drops, but in the secondary response, it stays higher longer. So you could say the response is longer the second time around. So one way that I remember this, if, for example, I had to answer a essay question that said explain 2 or 3 differences between the primary and secondary response, I remember faster, stronger, longer. That's what I remember. Now, of course, if something said explain the differences between the primary and secondary immune response, I couldn't say the secondary immune response is faster, stronger, longer. I could, but that doesn't explain anything. This is more a way to help me remember. It's a couple of words. It helps me remember. So a better answer would be the secondary immune response is faster at producing antibodies than the primary immune response. And I could add more if I wanted. I could say the primary immune response takes 3 to 6 days to produce antibodies. The secondary takes hours. And then I could say the secondary immune response produces more antibodies than the primary. And the secondary immune response, in the secondary immune response, the antibody concentration stays higher longer as compared to the primary. So very important distinctions. So let's move on. You do acquire immunity, and remember we said immunity is the ability to resist an infection or a disease. Okay, the ability to resist an infection or disease. So there are two types of immunity. There is naturally acquired, and artificially acquired. Naturally acquired is when you meet the pathogen in nature. So you're out and about, walking through the neighborhood, visiting with people. Artificially acquired is when you are introduced to the pathogen on purpose by a human, preferably a doctor or nurse. So you're introduced to the pathogen on purpose. Okay, doctor or nurse, hopefully. So that's the difference. Artificial, humans give it to you on purpose, usually in the form of an injection or a vaccine. Naturally acquired, you're just out and about and you meet it. Now, under each of these, there's two different types. So active and passive. Active means your B cells actively make antibodies. And remember the abbreviation AB for antibodies. Your B cells actively make the antibody. They encounter the antigen and they make the antibody. Passive means your B cells do not make antibody. Instead, someone is going to give them to you, give you the antibodies. So let's go back again. Naturally acquired, you are visiting a neighbor. The neighbor has the flu. You're exposed to the flu, and your B cells actively make antibodies. Okay, that is naturally acquired active. And the B cells might produce enough antibody that you never even notice you're infected. It might fight it off before symptoms, or it might not. Now, passive is different. In passive, your B cells do not meet the antigen, and they don't make antibodies. Instead, someone gives them to you, and the person who gives them to you is your mother. So the antibodies are going to pass from the mother to the fetus, either through the placenta or through the milk, through breastfeeding. There are different types of antibodies that pass through the placenta, then pass through the milk, but in either case, they both convey immunity to the fetus until the fetus's immune system kicks in, which is why it's one of the positive things about breastfeeding. Let's go look at artificially acquired now. So this is when you go to the doctor and they give you an injection, a vaccine. So active means, again, your B cells actively make antibody. So the vaccine they give you can be the dead virus, or it can be attenuated virus. Attenuated means weakened. So it's not dead, but it's weakened so that it probably won't give you any symptoms of the disease. It shouldn't. Most vaccines, by the way, are for viruses. It's harder to make vaccines for bacteria. So here's one way. You're going to be exposed to the pathogen, but someone is giving it to you in a vaccine. And it's either a dead virus, it could be a piece of the dead virus, or it could be a weakened virus. So some examples of live attenuated vaccines would be the measles vaccine, the mumps vaccine, chickenpox, yellow fever. There is a vaccine for tuberculosis, but that's a live bacteria. It is more difficult to produce these vaccines from bacteria. However, the live attenuated vaccines are probably the closest thing to the natural pathogen. So you get a strong cell-mediated and antibody-mediated response. So again, these kind of vaccines, when the virus is alive but it's weakened, kick your immune system in the most strongly to produce a really strong response, and they're really good. An example of a dead vaccine would be that for rabies. The rabies vaccine contains the whole rabies virus, but it's killed. The influenza vaccine is again the whole virus, or could be broken down pieces of it, but it is dead. Hepatitis A is another example of that. So these elicit a weaker response, so often you need additional shots or what they call booster shots. Now, what about a naturally acquired but passive exposure? Well, that's when someone injects you with the antibodies themselves. And remember, antibodies are called gamma globulins. So when would they do that? Well, they're going to do this when we cannot wait for your B cells to make the antibodies. Now remember, if it's active, your B cells are going to make antibodies, but that can take a while. With passive, you can't wait for your B cells to make the antibodies because you'll be dead by then. And an example would be snake venom, for an example, or botulism from the Clostridium botulism bacteria. You can't wait for your body to kick in antibodies. You'll be dead by then. So they actually inject you with the antibodies. RhoGAM, if you remember the chapter on blood, RhoGAM is the antibodies against the Rh factor, and that's injected into moms. So that would be another example. Okay, now this next slide, this last one, is really to show you how your immune system, both specific and non-specific defense, work together against a common enemy, against one particular antigen. So you can see here the x-axis is time in weeks, and the y-axis is the number of different immune cells that are active. So here is where you're exposed to a harmful bacteria, and the first one on the scene is the neutrophils, which is non-specific. Second one on the scene, natural killer cells, which is non-specific. Third one on the scene is cytotoxic T cells, which are part of your specific defense. We have macrophages, and then you see the blue line here, plasma cells, which are made by the B lymphocytes, and then you'll start to see the antibody being produced. So at any point in time, there are multiple components of your immune system, both specific and non-specific, attacking the same antigen, which is why your immune system is so powerful. I'm just going to end this with a video, which I'm going to let you watch, but this is a great video about why you feel sick when you are actually sick. What makes you feel sick? And you might be surprised to see that it's not the pathogen, it's your immune system response to the pathogen.